All right. So the Open Genome Project, as we know, was a project, an initiative that was actually funded by TIA. From as early as 2018, I became involved as a project manager in 2019, and it was conducted at Stellenbosch Medical School, and it was sort of like a very open project. We knew what the goals of the end goal of the project was. We wanted to implement ways in the clinic, and we wanted to sort of find a way to create these semi-automated, very comprehensive and comprehensible whole exome sequencing reports that can be used to implement personalized medicine. And but we didn't sort of know from the beginning how we're going to accomplish that. And that's what made it such an interesting project, because the goals, sort of the objectives, there were various objectives that shifted, that has evolved, and that eventually enabled us to reach our goal. And the goal was then to merge research, to back up what we want to do in clinical practice with ongoing research that feeds into that, and that identifies all the challenges and addresses it as we come across those challenges, so that we can come out with a process that will enable us to implement genomics in clinical practice, um, on a sort of multidisciplinary scale. So what we wanted to do from the beginning is make it innovative by merging social and technological innovation. And this is through the app that was created to sort of ease the process of informed consent and sort of capturing comprehensive patient information that could feed into the sort of platform to be integrated with the genetics, and then be outputted in a report that is meaningful. And the other innovation was the point of care technology that also came along to kind of help us to put together all that fragmented information and collect everything and serve as a validation step for extended testing. It was obviously patient-centric, as everything that we do is. And the app also helped us to make it accessible to engage patients and doctors, and to simplify sort of the intake process, um, um, before the genetic testing takes place. Obviously, we've been doing cutting edge research with all these innovations, and the innovation actually lies with the integration of all these different facets of genetics, like the pharmacogenomics, and that is focused on the medication side effects and separating or making a differential diagnosis between lifestyle related and familial disease, and offering the appropriate genetic test, and also relating that information sort of separately, but in the same, bringing that together in the same report. And other people all do that separately. We bring it together in a single report. And that we've only gotten to refine through combining research with service delivery. And so the milestones, the Open Genome Project had 4 milestones. The first one was the preparatory phase, because we really didn't know how we were going to do this in the beginning. And we thought the best way was to start to put together a team of people that would form the scientific and technical advisory panel. And these um, consisted of people such as yourself, so people from different sectors, clinicians, pathologists, scientists, genetic counselors, representatives of medical schemes, and so forth. And then we forged collaborations through this step where we could have our testing validated as part of the process, like, for example, in the NHLS, with their panels against our ways. And we also, in the process, developed the ethical framework for data transfer between different institutions, like, for example, between us and the MRC, all that ways and that whole genome sequencing data. The second milestone was then making sure that this platform, which is actually the Open Genome platform, which used the genomics sort of online integration platform. We needed to make sure that it was sort of technically competent to handle all the data that was going to be inputted, the patient data, all the algorithms that was built, um, to analyze the data and then to put it out into semi-automated reports. So this was a largely technical component that sort of was overseen in the second milestone. And from there, we then developed the pre-screen algorithm to select the appropriate patients for whole exome sequencing. And also then part of that was validating the gene variants that we found with extended testing with the gold standard, which is Sanger sequencing. So that was just getting sort of all the getting the tick marks for all the sort of technical competencies for the platform to make sure that it can handle all the data, that the data is secured, and that we have all the frameworks in place to be able to make that data available to the important people like the clinicians and our collaborators. Milestone 3 was the validation phase. And here we use point of care testing to sort of validate or sort of check the accuracy of voice, because we have seen that although we're working with sophisticated technology, we're working with sophisticated professionals, wherever there's people involved, you can have human error. And we've actually experienced that 2 or 3 times. And then we actually realized that we can use the point of care to sort of compare it with West results and see if the genotypes match, just to make sure that we have the correct sample or the correct data. Very important. Then we also sort of validated, the process of putting together all these fragmented information, the clinical information, the history of the patient, the lifestyle information, the pathology, blood test results, and the genetic data, if we can put that into adaptable patient reports. And the report has evolved so much since when we started that it's become now so easy to read, comprehensible. All the information that the clinician needs is on the first page, and whatever other details they need is in the rest of the report. And I think that was just so beautifully integrated in that report that has been constantly evolving through this process. The final milestone that we actually met after we validated the processes, when we had the report and clinicians were actually using it and kind of piloting it in their practice, and we got their input and then revised the reports accordingly. After that was done, we then thought, okay, let us pilot this, let us collaborate with the medical scheme, and let us do a medical scheme pilot and see if we can actually roll this out in a real world clinical setting. And that's what we did with Prof. Mani. And from that project that was published already last year in October, I think, and from that pilot already emanated a second pilot with another medical scheme that we have done, and we also want to actually publish that. So because of all this amazing work that has been done and is sort of still ongoing, we wanted to not let the Open Genome Project die with all that amazing outcomes, but we actually thought of expanding it now to the Open Genome Program. This is what we want to take forward, and we want to take it from the implementation phase and start sort of doing pilots with more medical schemes. And we want to just really increase access in that way to genetic testing to the public. And we're experimenting with different ways of doing that. And the way how we are also enabled to do that is through the new grant that we've been awarded. Actually, Genomics and not the university has been awarded this grant, which is actually wonderful. It's called the Advancement of a Pharmacodiagnostics Platform for Point of Care Assay Validation towards Whole Genome Sequencing in Clinical Practice. So we want to use point of care testing as a first tier test to get people sort of into the system, to kind of put them into the right care pathway that may or may not end into whole genome sequencing. But with all the information that is needed and is in one place for that patient. And so that is just one of the outcomes, was the Momentum pilot, but also we got a lot of articles from that process. We hosted a number of oncogenomics meetings online, 26 to be exact, and out of that actually came about 10 publications, actually more than 10 publications. And the people who published and who were on the papers included clinicians, genetic counselors, pathologists, all the people that we've mentioned. So we've made already a significant contribution in our own fields and in our own oncogenomics practices. And these are the publications that's already been that we already have under the belt. The latest one being with Prof. Marnie de Klerk and myself and a few other people. And this kind of sets the stage for the following publication, or this kind of overarching goal that we want to accomplish with the Open Genome Project, which I'll get to in a moment. And what I want to say about this is that we're very grateful for Prof. Barney and for Narina for joining, and we are only actually going to offer authorship to the people that makes the time to attend these meetings. I think we're going to give another opportunity in about 2 weeks to a month's time for the other people because it was a bit short notice. But we'll be offering authorship to the people that makes the time to attend these planning meetings. And then anybody else whose transcripts we use, because we have that at our disposal, we will just acknowledge them in the papers. But you are the people that we are looking to co-author with. And so far, we already have 5 potential publications that will emanate from this research grant. Maybe Prof can mention in more detail what they are. And we're aiming to actually get about at least 2 more publications out of this process. And what we want to address, what we've started addressing in the Open Genome Project and want to further address in the Open Genome Program is the pre-analytical, analytical, and post-analytical challenges related to the clinical implementation of WES. And the pre-analytical refers to clinician referral for genetic testing and the lack of actually recognizing who's eligible for testing, selecting the right tests and the right methodologies, and then ensuring that appropriate data is collected from the start and appropriate consent is collected from the start so that you have that data that will be integrated with the genetic data to give you meaningful results that can be used for management. But if you do extensive tests like whole exome or whole genome sequencing and you don't have, or any form of NGS, and you don't have all the clinical information, you cannot interpret accurately or adequately those results. So that is it. So that is one major problem that we have in here. We want to include the clinicians to kind of help us address this challenge in that 2 articles. Then you get to the analytical challenge, where that sets the stage. If you have problems here with collecting the data and selecting the right patients and the right tests, that sets the stage for the problems you face in the next phase, which is the analytical challenges, where the pathologists and the scientists have to actually integrate that data and put it into meaningful and actionable reports. And then the post-analytic challenge is that doctors are not yet confident or knowledgeable enough to actually interpret and actually report that complex genetic information to their patients. And we as genetic counselors are there to see that we convey the information accurately and clearly and prevent misinterpretation of the results and ensure proper follow-up and patient management, but we are also few. So there's a gap there that needs to be addressed with kind of the education of healthcare professionals as well in delivering those kind of results. All right, so what we want to do with this initiative is sort of have 3 focus groups. We have that focuses on the pre-analytic, that's the clinicians, the doctors, also kind of genetic counselors fits in there as well. The analytic group is the pathologists, medical scientists, and researchers, and the post-analytic is mainly the genetic counselors as part of a multidisciplinary team. And what we want to do is host about 1 or 2 more of these kind of meetings, but be more hands-on and kind of we already have an idea of what we want to do, the issues that we want to address, or the goal that we want to reach for the publication, but we need to figure out how every other author's specific contribution, what transcripts we're going to use, what information are we going to draw from it to sort of build out our papers. And so that's the purpose of these collaborative sessions, is so that we can have these discussions and refine those questions that we will address in the papers to reach our goals. The timelines are as follows. So phase 1 is already complete. We've invited authors. This is our first group meeting. And yes, it was a bit short notice, so we're hosting another one. The second phase is from March to sort of mid-April. We will host 2 more of these workshops, and then we will have started on the articles already. The ones that are in progress, we will start to finish them, and then the 1 or 2 more papers that we hope to start soon will be discussed as we go along. And then phase 3 in April. Between April and May, we want to start finalizing the manuscripts. And sometimes these are the only way to kind of get it out is if you give yourself a deadline and you work relentlessly towards it. So we would like to actually have submitted the papers by May month, because that is also the stipulations of the writing grant. So what is the main, like, where to from here? What are the next steps that we want to address in the next paper? So we want to focus on a core issue that Prof mentioned before the start of the meeting, and that's sort of the things like the inadequate coordination amongst healthcare professionals, and that we don't work seamlessly together with doctors' practices, and with medical schemes. There's this fragmentation of services. Also, that medical scientist doesn't have tariff codes for genetic testing, and there's also no kind of unethical way, or there's no ethical way really of getting doctors to do genetic tests on our behalf. There are companies that are giving medical doctors kickbacks. So they're actually starting to sell the genetic tests and the kits to doctors who then request the test because they get a kickback. I don't know how ethical that is, but these are the things that we are left to resort to when we don't have the structures in place to actually order the tests ourselves. So one huge requirement is for codes for medical scientists to order tests, because at the moment we need to kind of apply for a tariff code and go through a process to get a tariff code for each individual test. And that results in a lot of admin and also a lot of time wasting. So what we want to do is capture all of these issues that has already been mentioned in the paper published by Prof. Mani last year. And we want to take it further now, but sort of we want to be more intentional. We want to take the quotes from clinicians specifically, and we also want to invite the broader STAP group and show them that we have a whole group of people that are standing behind the initiative to obtain medical scheme payment for medical tests. And we want to use the quotes that people used in their talks and put it together and put this in the article so that that can also be something that we can use in this effort. So ultimately, we want to change current practice through highlighting these issues, getting everybody on board in support of our goal. And then we also want to discuss things like, do we need to invent a new tariff code for point of care? Can there be a universal code for genotyping versus sequencing or NGS? These are all the kind of things that we will get together and discuss. But yeah, that is just sort of an idea of where we want to go with this. And I think both of your input and your contribution will be invaluable to helping us make this happen. Thank you so much.